Mutations. Mutations are changes in the DNA, a change in the genetic materials. Mutations may be neutral, beneficial, or harmful. Most mutations are often silent due to the degeneracy of the code. What was the degeneracy of the code? So if you said it's when more than one amino acid is coded for more than one codon, you are correct. Types of mutation. Base substitution or point mutations. Base substitution or point mutations are the most common type of mutation. It occurs when a single nucleotide is replaced with a different nucleotide. So there's a change in one of the bases. There are three types of point mutations. Missense mutation is a base substitution that results in the change in amino acid. So it changes the codon and codes for a different amino acid. This usually results in a change of the activity of the protein. The change may be harmful or beneficial to the protein. Nonsense mutation is a base substitution that results in a nonsense codon. So it changes the codon and codes for a stop codon. This results in premature termination of translation. The protein is shorter than usual and does not contain all the amino acids that it should. Therefore, this protein is most likely non-functional. Silent mutation causes no change in the activity of the protein. So it changes the codon but codes for the same amino acid. Because the genetic code is degenerate, the resulting new codon may still code for the same amino acid. Frameshift mutation is caused by the insertion or deletion of a base pair. So you have the insertion or deletion of one or more nucleotide pairs causing a shift. This often involves a long stretch of altered amino acids, leaving a non-functional protein. So here's a picture of a normal DNA molecule. Here's the DNA strand. When we transcribe, we create an mRNA strand, and when we translate, here is our protein sequence. Now, in a missense mutation, as you notice here, we have AAA, and then we change the C to a T. By doing that, changing this T, we're now coding for changing the nucleotide to an A. So we have methionine, lysine, phenylalanine. Now we're no longer going to be coding for glycine. We're now coding for something else, a whole different protein. Same example with our normal DNA. However, we normally would code here TAC, TTC. But we take the T and we change it to an A. And that A then codes for U. UAG, that codes for a stop codon. We have now prematurely stopped this protein from being made. 9 times out of 10, 100% I should say, it is going to be non-functional. Say the same example here, and we'll see a frame shift in which we have AAA and we decide to remove that A. So now it's going to read AAC, CGA. So we're seeing a shift. So now it's going to read leucine alanine. So it's a total different protein. Mutations can result from the action of physical and chemical agents known as mutagens. So mutagens are agents that cause mutations. Spontaneous mutations occur in the absence of a mutagen. Chemical mutagens are nucleoside analogs. Nucleoside analogs are compounds that are similar to one of the 4 DNA bases but have different pairing properties. Let's take a look at, for example, 5-bromouracil. 5-bromouracil is the analog of thymine, but it sometimes pairs with cytosine. So thymine is going to pair with cytosine, and we know that's not correct. So T does not pair with C. In the picture here, it shows you 2-aminopurine. 2-aminopurine is the analog of adenine, but it sometimes pairs with cytosine. So adenine is pairing with C, which we know that also is incorrect. This is how AZT works to treat HIV. It causes incorrect base pairing for HIV, so it doesn't work correctly. Radiation. Ionizing radiation, for example X-rays and gamma rays, can cause the formation of ions that react with nucleotides and the deoxyribose phosphate backbone. Non-ionizing radiation is UV light, and this causes thymine dimers, which leads to the problems with replication and transcription. Bacteria have enzymes to fix the damage created by UV light. So they have what we call the nucleotide excision repair, which allows them to repair mutations if we do not expose them for the proper amount of time to UV light. Okay? So if we take a look here, we'll see that this bacterial DNA has been exposed to UV light, which causes the buckling, and thymine dimers are produced. When thymine dimers are produced and not all of the bacteria is killed, an enzyme could come and cut the DNA at 2 points and remove the damaged portion. So you have an enzyme, don't worry about the name of the enzyme, that cuts the DNA at 2 points and then removes the damaged portion. Now, DNA polymerase can synthesize a new DNA segment using the healthy strand. So remember, this was the healthy strand here as a template. Then DNA ligase can join the new fragments to the old strand. Gene transfer. There are 2 types. Vertical gene transfer. Vertical gene transfer occurs during reproduction between the generation of cells. It's when genes from parents are passed to the offspring. There's a passage of the entire chromosome in this event, and replication must occur first, then binary fission. All organisms do this, including us. So we always have a parent cell, and then we always end up with 2 daughter cells. Okay? Horizontal gene transfer is the transfer of genes between cells of the same generation. So genes to others in the same generation, sister to sister. So here is 2 daughter cells. They're going to be able to pass the DNA or genes to each other, like you passing your genes from your brother to your sister. And of course we can't do that. So only some DNA is transferred, never a whole chromosome, and it's not always an entire gene. Prokaryotes, again, are the only ones that can do this. So what are the 2 prokaryotes that can do this? If you said bacteria and archaea, you are correct. Mechanisms for horizontal gene transfer, conjugation, transformation, and transduction. Recombination. What happens to the DNA after it's transferred? Is it expressed? It is only expressed if it meets one of the following criteria. It is in a plasmid form, which is the method of transfer with conjugation, and it recombines with the recipient's chromosome through a process called recombination. Recombination is the exchange of genes between 2 DNA molecules. It results in the combination of genes on the chromosome. It occurs naturally within a genome and occurs with DNA transferred by conjugation, transformation, and transduction. So here we see we have a pink and a purple. Donor is purple, recipient is pink. we're going to have a crossing over that occurs when the two chromosomes are going to break and rejoin. So there's a crossing over, the two chromosomes break and rejoin, and we'll see that we now have a combination of the genes on each one. So the purple has some of the pink, and the pink has some of the purple. Donor DNA is transferred to the recipient cell in 3 possible ways. In transformation, the donor DNA is free in the environment. In transduction, the donor DNA transfer is mediated by a virus. In conjugation, the donor DNA is transferred by cell-to-cell contact. Let's start with transformation first. Transformation was discovered or demonstrated by Griffin. So Griffin's experiment was demonstrating genetic transformation, and he used a bacterium called Streptococcus pneumoniae. Streptococcus pneumoniae, when it has a capsule, causes pneumonia. Okay? So here is the live bacteria, and that live bacteria has a capsule on it. And the capsule, encapsulated bacteria, is injected into a mouse. That mouse dies. We take a sample from that mouse and put it onto a plate to create a pure culture. We can remove the capsule. Remember one of the things we said, when an organism does not have its capsule, then it's considered to be non-virulent. It cannot cause disease. So here we see that we have a non-encapsulated Streptococcus pneumoniae. We inject it into the mice. Nothing happens to the mouse. It lives. So as you can see, it's very different on pure culture plate. Now, what if we took that capsule, encapsulated bacteria, and heated it, and then injected it into the bacteria? Well, we know that most pathogens are mesophiles, and if we heat them, they die, right? Their proteins become denatured, and nothing is going to grow on the plate. However, if I took the heated encapsulated bacteria and put it with a non-encapsulated bacteria, we noticed that the mouse died because there was a transformation that occurred inside. So that means that these guys, these non-encapsulated strains, acquired the capsule. So let's see how that happened. Okay, so here's my donor DNA. Here is my recipient cell. So after death or when a cell lyses, some bacteria releases their DNA into the environment. So when we heated that encapsulated bacteria, there was a release of the DNA into the environment. Other bacteria, generally of the same species, can come into contact with these fragments and take them up. So the recipient DNA takes up the donor DNA through its cell membrane. So remember, these are lowercase, these are uppercase. And the donor DNA is going to be taken up through the cell, into the recipient cell. And from there, it's going to incorporate them into their DNA by recombination. So the DNA must recombine to notice a possible change in the genotype. So here's the lowercase and the uppercase. Remember, we have to nick it, have a crossing over event that occurs, and look at that. We now see that we have some of the lowercase, which is the lowercase a, that is now a part of the recipient DNA. Recombination, again, is necessary for the expression. So this would explain why the mouse died. The pieces of the encapsulated DNA was taken up, and now this cell now expresses a capsule. Conjugation. This mechanism requires the presence of a special plasmid called the F plasmid. The F plasmid facilitates conjugation. Bacteria that have F plasmid are referred to as F positive. So here you go. So if you have the F plasmid, here's the F plasmid. That is a male cell. So F positive or male. Those that do not have the F plasmid are F negative cells, and they are female. So male, female. The F plasmid consists of 25 genes that mostly encode for the production of the sex pili. A conjugation event occurs when the male cell extends his sex pili and attaches to the female. So the male cell is going to extend his sex pili and attach to the female. This attached pilus is a temporary cytoplasmic bridge through which a replicating plasmid is transferred from the male to the female. So here we'll see that we have our F plasmid. Through Rowland circle replication, okay, we're going to see that this is going to be transferred to the female cell through the sex pilus. Okay? When the transfer is complete, the result is going to be 2 male cells. So there's a sex change. So now that this cell has a plasmid, this is also known as a male cell. And here's a picture showing you the F positive cell and the F minus cell and the sex pilus between them. Transduction. In transduction, you have a bacteriophage. A bacteriophage is a virus that infects bacterial cells. A phage attaches to the cell wall of a bacterium and injects its DNA. So the phage injects the DNA into the host. The bacterial chromosome is broken down, so the chromosomal DNA of the host is chopped up. Meanwhile, biosynthesis of phage DNA and protein occurs. So the phage forces the host to make phage DNA and phage proteins. So we chopped up the chromosomal DNA of the bacterial cell, and the bacterial cell is now forced to make some of the phage DNA and proteins. The phage protein and capsid surrounds the pieces of the DNA to make new phage offspring. Well, sometimes when it's doing that, it's packaging some of the bacterial chromosome DNA. Do you see that? Because the pink represents the phage DNA, the purple represents the bacterial DNA, and it's packaging some of the bacterial DNA. And when it does that, it's going to be released from the host by lysing it. So the cell lyses, releasing some of the viruses. And of course, it's like, ooh, look, next door, there's another bacterial cell. The normal offspring phage are released, and they go ahead and they infect a new host. But what you see here is you have a virus that has the bacterial chromosome from the other bacterial cell, and it now goes into another bacterial cell, and recombination occurs. And check this out. Now, this DNA is now inside of this one, so this cell now has a whole new genotype and possibly phenotype. Here ends Chapter 5.